All right, welcome to the videos for Chapter 6. This is the start of Unit 3. In this section, we're going to focus a lot on bacterial growth, what they need for that growth, so nutrition. And then at the end, we'll talk a little bit about this process called differentiation. So bacteria tend to divide symmetrically. So we start with one and we end up with two that are pretty much identical. But there are some cases where bacteria can make some different structures, and that process is called differentiation. Now, your body, your cells, you start as a single cell, right? Sperm and egg fuse together and become the zygote. That one cell divides in different ways to become all the different cells that are in your body. So you have liver cells, you have skin cells, eyeball cells, brain cells. They're all different, and they get that way through the process of differentiation. So that's what that word means. This chapter is going to tie in very well with the lab. We're going to do some of the procedures you see here in the lab, so really good to get a grip on them here. And we'll come back to some of these concepts when we talk about metabolism, so particularly growth and nutrition. How does that tie into metabolism? And you might think, why do I need to know about how bacteria grow, right? What does that matter? Well, it's really important for identifying bacteria, but also because we are evolutionarily related, bacteria and us, we do a lot of the same metabolism processes. So if you are taking this before A&P or something like that, the metabolism you learn for bacteria will be very similar to your own. So in this chapter, we're going to talk about nutrition and how the environment can impact the growth and differentiation of microbes. We'll talk about a bit about how microbes are classified based on these nutritional needs. We're not going to get into like this genus and this species needs this, but we'll talk about words that describe the ways different microbes need certain nutrition to live and grow and divide. Then we'll talk about how understanding this growth helps us identify disease-causing microbes, pathogens. And finally, we'll talk about the structure called a biofilm and why it's very important for the infectious disease processes. So understanding them is quite important. This chapter is kind of about knowing your enemy, right? Knowing how it works, but not just that, right? Not all microbes are pathogenic. We're also going to talk about how microbes interact with our biosphere as a whole. How do they play critical roles in cycling carbon and nitrogen through the environment? Without microbes, we could not survive. So they're not just bad things. We have ones that we need for our own survival. All right, so we're going to start with a little case study, and we'll see a few other ones smattered through this chapter because a lot of these tie directly into the concepts that we're going to be talking about in this chapter. So we have a young mother who recently emigrated from Uganda and brought her 18-month-old infant into an ER room in New York City. So the infant's crying and in pain. That's pretty obvious. And the problem about infants is you can't say, where does it hurt? So the physician is going to kind of move them around, try and see what happens. And when they move the baby's head, that's where we notice a lot of crying and pain happening. So the first thing the physician is going to suspect is meningitis. And we've already seen a little bit about meningitis. By the end of this course, we will have so many examples of meningitis, you will know it quite well. So meningitis is a bacterial or microbial infection of the cerebrospinal fluid, and that leads up to the brain with the meningeal layer. So that's not a great place that you want to have microbes growing. When you have this infection, your body often hurts when you move your head because you're moving your spinal cord in this. So it's the spinal fluid that's in there that gets infected. So they're going to do a spinal tap where you collect cerebrospinal fluid, and normally this is a sterile fluid. We saw a previous example where we saw that it was cloudy. So they would probably see that it's cloudy in this example, but we want to know what's in there because we need to prescribe the proper drugs to treat this. So you can look at it under a microscope, and we've done this in the lab now, and we will do the gram stain as well. So under the microscope, we see a gram-negative bacilli. That tells us something, but it doesn't really narrow it down. There's a lot of gram-negative bacilli. It could be E. coli. It could be many, many things. So what we're going to do is we're actually going to grow it on what we call solid media. This is our petri dish, right, with some sort of growth medium in the bottom. And you can see these two are different. This one's a red one, and this one's kind of a brown one. That has to do with the different nutrients and things that are in these plates here. And this is going to tell us how the organism grows on these things. That'll give us clues as to what it is, because some microbes don't like one versus the other. So the organism, whatever it is in there, gram-negative bacilli, doesn't grow on this red plate, which contains whole intact red blood cells. It's called a blood agar plate. But it did grow on this one with lysed or broken red blood cells. We call this chocolate agar, not because there's chocolate in there, but because it's chocolate colored. So it grows on this and on a media containing what are called growth factors. And they have very creative names like X and V. We'll talk about these a little bit later, but basically they're additives that some microbes need to grow. So if they need this, it tells us certain things about them. This is an example of kind of diagnostic plating, right? We did a diagnostic when we looked at it under the microscope. This growth pattern is another diagnostic that can tell us something. The diagnosis, based on what we call a metabolic pattern, is that this is likely Haemophilus influenza, which is a bacteria. It sounds like it causes the flu, but it doesn't. It causes symptoms that are similar to it. The flu is actually a virus, not a bacteria. So in this case, because it is a bacterial infection, we can prescribe antibiotics, and the child makes a full recovery quite rapidly. But what's going on here? Well, this Haemophilus influenza is a specific strain called strain B. It's particularly invasive and can get in through contact or through our airborne routes. And there is a vaccine for this that was developed in the late 80s. So I probably would have got this as a baby. Prior to this vaccination, there were a lot of cases of childhood meningitis, even in developed countries. So this would have been very common. In this case, we have someone who's emigrated from a foreign country where they don't have access to these vaccinations. So their child was easily susceptible to this. So this is one illustration of how childhood vaccination can drop the numbers of these easily prevented infections. And ultimately, this could have killed the child. So in this case, we were lucky and we were able to deal with the infection with antibiotics, but getting vaccinated is very, very important for people who come here and people who have children here. Okay, so we used some techniques here, and you can start to see, right, we had to grow bacteria in there to figure out what they like, what they don't like, what they can use metabolically. And so we're going to talk a bit in 6.1 about this technique called culturing, which is just a fancy word for growing microbes. And we can use this to do some different processes. One of those is counting the number of bacteria, because most things are not completely free of bacteria. There's always a few floating around. But the level of bacteria, how much is in there, like in your blood or in your gut, the level matters. When you have a particularly bad infection, usually the level of bacteria goes up, and the higher it is, the more worried we are and the different steps we have to take. So sometimes we want to actually count how many bacteria are in some sort of substance or sample. So we're going to talk about this process of obtaining a pure culture. So remember back to Koch's postulates. He needed a pure culture, which is just one single species of microbe to show that chain of infection. So we're going to learn how to do this, and we'll do that in the lab as well. It's simpler than you think. We're going to talk about different growth mediums, right? So that's things to grow bacteria in and on. So we'll talk about the different growth mediums. It's really pretty simple, the level we're going to go to. And then we'll talk about ways to describe bacterial growth how it's measured, and talk about the advantages or disadvantages of some of the methods. We're not going to go into an exhaustive amount. If you're really interested in how we can count microbes, there's a whole bunch of extra ones in the book that are pretty cool. So this is really critical for diagnostic microbiology because growing things on plates like we just saw in this example is one of the cheapest and quickest ways to identify what's there. Okay, so growing bacteria in a culture. Microbes naturally exist in the environment, and usually they're in communities that have multiple species in there. But for lab studies, we need to grow the species separately. So we take a sample, I don't know, you take a stool sample, which is a poo sample, right? There's going to be a lot of different bacteria in there, even in a healthy individual. What if we want to separate out those bacteria? If we want to get the individual species, that would be pure culturing, so getting a single species. And to do that, we need to use specific techniques. Now, let's back up and talk about the two ways that we can grow bacteria in the lab. We have liquid media. This is what it sounds like. It's a liquid medium. Usually it's got some kind of nutrients, and sometimes it's called broth or something like this. That's not like chicken broth from the store, but it has some of the same additives. It might be yeast extract or things like that. It's got nutrients in it, right? And this is great for looking at things like growth rates. How quickly do bacteria grow and divide? But it's not great for getting a pure culture because everything's mixed up. The other type of medium for growing things is solid media. This is great for separating mixtures of different organisms. So you can see here on this plate, there's all kinds of different things. We've got this pink one here, this orange one, a yellow one, bright yellow one, a white kind of fried egg looking one, these little dot ones. There's all these different species here. Each of these dots is what we call a colony, and that colony started as a single bacteria, and it divided by clonal reproduction or binary fission into this colony here. So if I took a little bit from this colony here, they would all be genetically identical because they started from one single bacterial cell. So these two methods, they both grow, but they're used for different things, and we'll talk about each now. Okay, so say we want to take our mixed sample, right, our stool sample from an individual, and we want to get pure cultures of just one type of bacteria. Well, we're going to use a solid medium, as we said, so an agar plate, and we want to get colonies, right? Each of these dots, again, is a colony. You can see over here, it's really, really thick. The colonies are all smushed together. That's not great. But out here, they're nice and separated. So we know that that colony is started as a single cell and should be a single species then because of that. So to get this plate that looks like this, we do a technique called isolation streaking. So this is streaking to isolate, right? It's right there in the name. And it's to get these individual colonies, to get pure cultures. So what you do is you start with an inoculating loop and you get your sample, whatever it is, and then you do 3 different streaks. So we dip into our sample, we do a streak. Then we flame our inoculating loop to sterilize it, let it cool, and we pass through a little bit of this to do our second streak. So that's going to reduce the amount of bacteria, right? We're just dragging a little bit of it. So the first streak is really thick. The second one gets less thick. If we reflame our loop again and do a final series of streaks, we should have separated out and get these individual colonies from that. So this is isolation streaking. Super useful. Okay, we're going to watch an animation of this one because it's easy to see it in practice.
Agar medium in a petri dish provides a large flat surface on which a mixture of microorganisms can be streaked to separate individual cells. In the dilution streaking technique, an inoculating loop is sterilized in a flame and allowed to cool. The loop is used to collect a drop of liquid from a bacterial culture. With the loop held flat on the agar, The drop of liquid is streaked across a region of the agar plate's surface. As the loop moves, organisms begin to rub off the loop. The loop is sterilized and cooled again. This time, the loop streaks over the first pass, collecting a small number of bacteria and spreading them into a fresh area of the dish. The procedure is repeated again. Toward the end of the streak, few bacteria remain on the loop, so at that point, individual cells will land and stick to different places on the agar surface. If the medium contains the proper nutrients and growth factors, a single cell will multiply into many millions of offspring, forming a colony. In general, a pure culture can now be obtained by touching a single colony with a sterile loop and inserting that loop into fresh liquid medium. It is important to note that the one cell equals one colony paradigm does not hold for all bacteria. Organisms such as streptococci or staphylococci usually occur as chains or clusters of several cells. A cluster of 10 or so cells called a colony-forming unit, or CFU, may give rise to a single colony.
So that's an isolation streak. That's for getting pure cultures, isolating bacteria. What if we want to count bacteria? Well, to do this, we need to use a process called dilution, and we'll cover this again in the lab, but if you're not familiar with the process of dilution, let's talk about it. So, if you've ever gone to the store and you get the frozen can of what's called concentrated orange juice or something like that, it's a big frozen log of super dense concentrated orange juice. And you dump that out into a container and then you add water to it. Put it in the thing, and as you add water, you're diluting it to a lower concentration that actually tastes good. So, dilution is just like that, but in this case, we're diluting our bacteria. So we have a flask of bacteria here that is grown to a certain, what we call density, a certain number of cells per milliliter here. So in this case, I have 10 milliliters of liquid in this flask, and for some reason, this is just a hypothetical, I know that there are exactly 1,000 bacteria in here, right? So if I were to take out 1 milliliter of this liquid, how many bacteria would I be taking out? Go ahead and pause the video, think about that for a moment. Okay, so some simple math, right? It's working in tens. I've kept it easy. We have 1,000 bacteria in there in 10 milliliters. So what's our concentration? It's 1,000 per 10. And we could simplify that down to, cancel the zeros, 100 bacteria per milliliter. So if I take out 1 milliliter, I should be bringing with it 100 bacterial cells. Now, this is speaking statistically. Of course, in reality, things aren't always mixed perfectly and stuff like that. But statistically, if I did this over and over again, I should get statistically 100 bacteria in 1 milliliter. Okay, cool. So that's not dilution per se. It's still at the same concentration. But what if I took that 1 milliliter and added it to 9 milliliters of fresh liquid medium? So I've taken my 100 bacteria in 1 milliliter and added it to 9. Okay, here's where it gets confusing. This dilution here is what we call a 1 to 10 dilution. Okay, I don't like that terminology. It's confusing. This is really 1 milliliter becomes 10 milliliters. So 1 plus 9 of fresh becomes 10 total milliliters. You can express this, right, as 1 over 10, or scientific notation, 10 to the minus 1 is the same as saying 1 over 10. Okay, so in here, we now have a total of 100 bacteria, but it's in, right, 10 total milliliters of liquid. So what's the concentration per milliliter now? Think about that. Pause the video. Okay, so we have 100 bacteria in 10 milliliters. Cancel the zeros. That's 10 bacteria per milliliter. So we've diluted it to a lower concentration. So what happens now if I take 1 milliliter out of my dilution? How many bacteria do we have in that 1 milliliter? Pause the video if you need to. We should have 10 in there, and we've put that into another 1 to 10 dilution, which becomes now a total of 1 to 100. So we did 1 to 10 and 1 to 10. You multiply those two together. Now you have a 1 to 100 dilution of the original sample. So statistically, in this 10 milliliters, we have 10 bacteria. What if we do this again? How many bacteria do we have this time? Another 1 to 10 dilution, which becomes a 1 to 1,000 or 10 to the minus 3 dilution. How many bacteria do we have in there? Statistically, we should have only 1 in there at this point. So we've diluted it down to lower the concentration each time. Now, why does that matter? So in reality, we wouldn't know the concentration of our starting bit, right? I told you it was 1,000. I just made that up, right? But in reality, we have this unknown concentration. But we want to figure out how many bacteria are in there. What's the concentration of it? How many cells per milliliter? To do that, we're going to do our series of 1 to 10 dilutions, which is called a serial dilution series. And we're going to take a small volume of each of these and plate them out. And using some math, it's not scary, I promise, we can determine the original concentration here. So we'll do an example here, and we'll actually do this in the lab. This process of spreading some of this diluted solution on the plate is called spread plating, because you literally spread it across the plate. We'll do that in the lab, but you use a little spreader and you spread it around on the plate. So it's called the spread plate technique, and this is great for getting bacterial concentration when you do it with this dilution series. This tenfold dilution series is pretty standard. We'll do that exact one, and you use 10 because it makes the math really easy to do there. So early on, you can see that it's really dense, right? You can see here that the culture is really dense, and as you start to dilute it, it becomes lighter and lighter because there's less cells in there. So if we spread 0.1 milliliters of this one here, 10 to the minus 3 in this case, there's too many bacteria on the plate to count, okay? They're all clumped together. We can't accurately count the colonies. But if we do the next one, 10 to the minus 4, okay? So 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, we can actually count that number. And we get 254 colonies on there. If we do another 1 to 10 dilution, 10 to the minus 5, we get 23 colonies on there. Do you see a relationship between these numbers here? If we do another 1 to 10, 10 to the minus 6, we get 3 colonies. Do you see the relationship between these numbers here? They should be decreasing by a factor of 10 each time. And you can see it doesn't work out perfectly here. This should be 2 colonies, not 3. But using these values, and when we do it in the lab, we will look for a range, I don't know, between 300 and 30 colonies-ish. This one we could work with, but as you go down, the statistical variation makes working backwards more error-prone. We can work backwards from these values and knowing our dilutions to multiply and figure out the original concentration of this flask. So this early plate, it has what's called confluent growth. We just always call it a lawn because it's like just completely covered with bacteria. So this is a lawn plate right here. And the important thing about this counting technique is each colony represented a what's called viable organism, or an organism that can reproduce in this original concentration. There might be dead cells in here, but we really want to know how many living cells or capable of reproducing cells are in here. So by growing them through this process, we're getting a viable organism count. Now, what if we wanted to know how many dead ones are in there? Well, there's different ways we can do that. I'll show you in a moment. Okay, so again, note that the dilution numbers are rarely perfect, right? If this is 254, this really should have been 25, and this should have been 2. Doesn't always work out quite that accurately, but we can work backwards from this to get the original concentration. Okay, so here's a real example. We have a dilution series, so I took a flask that had a solution in it. So I took a flask that was growing bacteria, and I want to know what the concentration in there is. So that's how many cells per milliliter is there in that flask. So I did a dilution 10 to the minus 1 here, right? That's a 1 to 10 dilution. 10 to the minus 2, 1 to 100, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, and 10 to the minus 6. You can see these first ones, this is a lawn. This is totally uncountable, or too numerous to count here. Same thing with the 10 to the minus 2, although you can see a few individuals. 10 to the minus 3 getting better. 10 to the minus 4, maybe if you had an hour to count this plate, but it's really, really dense. So we're going to rule this one out because some might be squished together. Now, when we get to 10 to the minus 5, you can count this. It might look tedious, and it is. You take a little pen and you put dots on there and you click it with a clicker. But somebody did it for us, and there are 107 colonies on here. 10 to the minus 6 has 11. So this relationship is close to a tenfold difference, right? So using this value, this dilution factor, as it's called, and this value, the number of colonies on here, we can work backwards with some math. 10 to the 5th, which is the inverse of 10 to the minus 5th, times 107 colonies equals 1.07 times 10 to the 7th colony-forming units per milliliter. This is 10 to the 7th, right, is 10,700,000 cells per milliliter. So there's 10,700,000 bacteria in every milliliter of culture liquid that's in this example. So, right, this is exactly what's happened here. I now know the concentration of this. And you might say, why do you want to know how many bacteria are in that flask? Well, it might not be a flask. We'll see a human example, a human medicine example in just a moment. So remember that term CFU, colony forming unit? They talked about it a bit in the animation. Well, some bacteria kind of clump together sometimes. So ideally, a colony started as one single cell, but some things like these staphylococci, they tend to move in clumps, so we can't be sure that each colony started as a single unit. It could have been 2 bacteria. So that's why we use this term colony-forming unit. You can think of it as cells per milliliter. It's the analog to it. Okay, so we've seen isolation, we've seen counting. Both of these required growth mediums to grow things in. And there are different terms to define different mediums. These are some kind of broad terms here. So a synthetic medium is not that it's artificial in any way. It's more that we have made it and precisely defined the contents of it, right? So we put X amount of iron and X amount of glucose and X amount of whatever chemical, phosphorus or nitrogen in there. So synthetic mediums are really highly defined, and this can be important if you're trying to grow just a very specific thing. If we take things back and only put in just what our microorganism needs to grow, this is called a minimal medium. It's a synthetic medium that has only the nutrients the organism needs to grow, nothing extra, right? Just what it needs. So it could be water, some salts, a carbon source like glucose, a bit of nitrogen, and some sort of energy source there. That could also be glucose. This is just what's in there. There's no extra things. It's the if you took one of these out, nothing would, the organism wouldn't grow on there, right? It needs all of these things, but it doesn't need anything else. This is really important when you're testing between two different bacteria, right? In that example at the beginning, we had the chocolate agar versus the blood agar, right? Being able to see that it grew on one versus the other, that was where this minimal media comes in. You can also just kind of say, meh, whatever. I'm going to put a lot of stuff in there. These are nutrient-rich mediums, but they're not super well defined. We're just like, yeah, throw a bit of this in there. We don't know exactly how much is in there. These are called complex mediums or rich mediums. This could be something like beef extract, which is kind of like ground up beef that's thrown into some other salty liquids, or yeast extract, ground up yeast. We don't know what's in there or how much is in there, but, sometimes we don't care. Sometimes bacteria can be picky eaters, though, right? Those of you that have children may have suffered from this. Bacteria are the same way. They like certain foods and they don't like other foods. Some bacteria need enrichment in their media. They have to have special nutritional requirements that get added to it beyond what's normally in a basic normal plate. And this is a great way to identify things, right? The fussy eaters won't grow on this one, but they'll grow on that one. Really a good way to tell the difference here. So how do we use this? These are two really important terms that we're going to come back to again in this lecture, and we'll do this in lab. We're going to talk about selective versus differential media. So using these media types, we can do two processes. We can select for specific organisms, and we can differentiate or tell the difference between different organisms. So let's talk about selective first. A selective medium has compounds in the media that prevent some types of bacteria from growing, favoring the growth of other types or one specific type. So TLDR, it lets some grow, but not others, right? That's selection, allowing some things to grow, but not others. That's what the term selection means. We might see this in an example where we want to grow gram-negatives but not gram-positive bacteria. We could use a selective media that selects for gram-negatives. Differential media, it allows species to grow equally well, but there are compounds in there that are metabolized differently and change something in there that can be distinguished. Generally, this is color. So look at this plate over here. You'll see some kind of pink-purple colonies, and then these little white colonies over here. This is differentiating between the two. The ones that are pink are bacteria that can ferment lactose sugar. The ones that are white cannot ferment lactose sugar. So we can differentiate between those two based on color here. So TLDR differentiates between two types by color change. So this plate here is both selective and differential. This is called McConkey medium. I would like you to know this example specifically. It's pretty simple. So in this plate, in this McConkey medium, there are what we call selective agents. These are just chemicals, bile salts and crystal violet, which will prevent the growth of bacteria except for gram-negatives. So gram-negatives will grow on this plate, but gram-positives, other types of bacteria, they won't grow on here because of these things here. And you might recognize crystal violet from the gram stain. The gram-positives, it goes into their cells and it actually inhibits them from growing because it gets stuck in there. So gram-negatives are selected for here. Now, we want to differentiate between different types of gram-negatives. In this case, we want to determine, can they ferment lactose or can they not? That's where our differential agent comes in, which is lactose. But when the bacteria ferment the lactose, if they can, it creates byproducts that change the pH, which makes this pink color here. We have an indicator dye in there, and we'll talk about this in detail in lab. But the pink is indicating that fermentation has occurred. If they can't ferment, they don't create those byproducts, and there's no pink color made from it, so they show up white. That would be differential. So this is both a selective and a differential plate. Now, like I said, this term selection, pretty common term. Where have you heard this maybe in biology before? Think about Charles Darwin here. You've probably heard of natural selection, right? Some organisms survive and reproduce, whereas others don't. Or it could be artificial selection. Our different dog breeds are all products of artificial selection, right? People who like, teacup poodles or something like that, selected for ones that stay really small throughout their life. So let's look at a real example using this McConkey plate here. Okay, we have a 15-year-old who goes into the ER. They have severe abdominal cramping, diarrhea for the past 2 days. This is where, being a sample collector, which you may very well be, is not very fun. You have to get a fecal sample, and that sample is sent to the lab then. It has to be collected properly and sent to the lab, and the lab tech is going to take that and streak some of that in this isolation streak on the McConkey medium. Okay, so why use McConkey? So gram negatives get selected for. Most of our gastrointestinal issues come from infections by gram-negative bacteria. Gram-positives don't tend to cause GI problems. They don't tend to cause diarrhea. So in that fecal sample, there's lots of bacteria, right? There's going to be all kinds of things in there. There might be gram-positives, but we want to rule all of those out. We're looking for a gram-negative that could be causing this issue. So the selection here, that gets rid of all those gram-positives and everything else. Now, we want to determine between the gram-negatives who should be there and who shouldn't. So in our normal gut microbiome, there are bacteria that have evolved with us. What do you do when you are a baby? What do you drink? You drink milk. And milk has lactose sugar in it. So the bacteria in our gut have evolved to metabolize lactose. Okay, they can use it. Those are the good bacteria. Generally, the bad bacteria that cause the diarrhea and things like that, they can't metabolize lactose because they don't normally live in your stomach of a milk-drinking organism. So in this example, the pink colonies, or the red colonies here, are E. coli. There's tons of E. coli in your gut all the time. It should be there. It's a non-harmful, non-pathogenic strain. The white colonies, though, they shouldn't be there. They don't ferment lactose, so they generally shouldn't be in our gut. Those are Salmonella enterica, which are causing, in this case, the diarrhea. Okay, so that's an example of a selective and differential plate. I teased this a bit. We want to know how much bacteria is in a sample. So let's take this case history. This is one where numbers actually matter. So we have a urine sample. Okay, we have Melissa, 25-year-old woman, complaining of a burning sensation while urinating. Shouldn't be happening, right? So the physician assistant suspects a UTI, urinary tract infection, right? From the signs and symptoms, that's what it seems like. But we want to confirm that is actually the case, that there's not something else going on. So we're going to take a urine sample. Again, not fun, but necessary. And the lab tech is going to take a small volume of that urine sample and do that dilution plate series, right? And then it's going to grow, and they're going to count the colonies and work backwards through the math. After 24 hours, the microbiologist found large numbers of E. coli colonies, that gram-negative bacillus, equivalent to 100,000 bacteria per milliliter in the patient's urine. Now, this is bad news because urine samples should normally have pretty much no bacteria. They're not quite fully sterile, but they should have very low, if any, bacteria in there. This is a very high number for a urine sample. So we can conclude that those E. coli that are in there, they're at a high enough number to cause that UTI. So they're going to prescribe antibiotics against the E. coli to solve that problem. So this is why these bacterial numbers are so critical. This counting is so important. And just looking at the urine, yeah, you could maybe see it's cloudy, but you really want to know the total number that's in there, because we would probably, if it was really, really high, prescribe intravenous antibiotics versus a pill if it's kind of moderately high. If it's really, really high, UTIs can be very dangerous, particularly to the elderly. So these are all methods of indirect counting, and they rely on growth, right? So they're viable organism counts. So in our urine example, they would have done the exact dilution series. They would have got, in this case, they spread 100 microliters, that's 0.1 milliliters, on the plate. If they got 100 colonies from 100 microliters, that's 1 cell per microliter, right? And you can do the math. If that dilution had been done to like 10 to the minus 3, then you could multiply by 10 to the 3, and we get our 100,000 cells per milliliter there. So this dilution series, we're going to do it in the lab. We'll come back to it. Don't freak out too much about the math. It's just factors of 10. Okay, those are indirect ways. We have some other indirect measures that don't rely on growth, right? So this relied on growing colonies. It's very accurate. Sometimes we need to go a little bit faster. Growth, even though it's fast with bacteria, it takes time. What if I want to know right away roughly how many bacteria are in something? To do this, we can use a special sensor called a spectrophotometer, which measures what is called optical density. Optical density is a fancy word for how cloudy is something. The cloudier it is, the more things are in there. So the spectrophotometer works by shining a light source through a prism, and then it shines it through our sample, and a certain amount of it, depending on how cloudy this is going to get absorbed. And we know how much went in, and then that means we know how much got absorbed because our detector tells us, and it reads out a number. So what does this mean for our counting example? Well, you can see, right, the original sample is super cloudy, and as it gets more dilute, or as there's less cells in there, it gets lighter and lighter. The spectrophotometer is basically just measuring how cloudy it is, and the simple mathematics here is more cloudy equals less light, more cloudy equals more cells, right? The problem with this is we can't tell if the cells are live or dead. Sometimes it doesn't really matter, but it's not as accurate as counting would be, because this is a viable organism count. There are cool techniques to directly count how many are alive or dead. This is a fluorescent microscope that has a stain that shows up green if the bacteria is alive and red if the bacteria is dead. Now, this would really suck to count by hand. You could maybe program a computer to count red versus green dots. That would be a direct count. Looking through a microscope and directly counting how many bacteria are there. There are some techniques to do that. There's things called hemocytometers that get used, but we're not going to talk too much about those. If you're really interested in that, there is more info in the book on that. Okay, so that was a lot, but super critical to understanding how bacteria get grown, how we count them, how we isolate them, because that's what all of microbiology is based on. So a pure culture, right, contains only a single bacterial species that is genetically identical. It started as one cell and grew into that colony. So we need these pure cultures for detailed research work, biochemical studies, and also to test what antibiotics those specific bacteria are susceptible to. So that's why we do this for individual patient samples. Bacteria can be cultured on solid or liquid medium. We have different types of medium with different definitions, right? Minimal mediums contain exactly only the nutrients essential for growth. then complex or rich ones have many, many more nutrients. You can look at the differences between organism growth. We have selective plates. Do they grow versus do they not grow? So we select for a specific type. And we can use special metabolism and things like that to differentiate between different types. So we have selective and differential plates. Some plates are selective and differential. Bacteria can be counted. They can be counted directly under a microscope, but they can also be counted indirectly through our viable counts, our dilution series and plating, or using that optical density, that spectrophotometer cloudiness measure, basically. I want to reiterate, a viable bacterial organism is defined as a cell with the ability to replicate and form a colony on there. So basically it means it's able to grow and divide. All right, that's it for 6.1.